Good morning, everyone. This is Sri Priya, and today my topic of presentation is Prokaryotic Translation, Regulation, and Proofreading of Prokaryotic Translation. Let's first see what is translation. The translation is the process in which a polypeptide chain is obtained from an mRNA strand. The mRNA has an ordered series of three nucleotides which are called as codons that codes for amino acids. These codons are present as non-overlapping series on the mRNA, which are called as open reading frames or ORFs. The translation process starts with the start codon and it ends with the stop codon, and it occurs in a 5' to 3' direction. And translation process in prokaryotes is polysystonic. What are the sites of translation? The sites of translation in prokaryotic cells is found to be in ribosomes of 70S type. This is further divided into two subunits, that is the larger subunit that is 50S type and smaller subunit that is 30S type. The larger subunit will in turn show 3 parts and smaller subunit will in turn show 2 parts. What are the requirements of translation? It requires a 70S type ribosome that in turn has 3 sites, that is A site, acceptor or amino acid site, the P site that is peptidine or donor site, and E site that is exit site. It also requires a mRNA that is the messenger RNA, a charged tRNA that is the tRNA that carries an amino acid, and certain factors like initiation factors, elongation factors, and release factors. It also requires a GTP molecule for energy and Mg2+ ions. What are the steps involved in prokaryotic translation? There are 3 main steps, which are initiation, elongation, and termination. But before initiation starts, the tRNA has to be charged, and it is done in 2 steps. The first step is activation of amino acid, and the second step is tRNA charging. In the first step, the amino acid will get combined with ATP and it forms amino acyl adenylate in the presence of an enzyme, amino acyl tRNA synthetase. Whereas in the second step, the produced amino acyl adenylate will then get combined with the tRNA in the presence of amino acyl tRNA synthetase, and it will produce a charged amino acyl tRNA. In this process, one AMP molecule is also released. Then comes the initiation step. When the initiation step carries out, initially the 30S and 50S subunits of ribosomes are not together and they are present as separated forms. Then, on the 30S subunit, the IF3 initiation factor will come and bind on the E side, that is the exit side. This will help in preventing the association of 50S subunit with the 30S, and also it blocks the P side. Then afterwards, the IF1 initiation factor will come and block the A site on the smaller subunit, which prevents the attachment of tRNA at the A site. This smaller subunit will then in turn get associated with the mRNA chain by base pairing between the ribosome binding site on the mRNA and 16S rRNA present on the smaller subunit. This ribosome binding site is also called as Shine-Dalgarro sequence, which is seen only in prokaryotic cells. This is the Shine-Dalgarro sequence of the prokaryotic cell, and this is the complementary 16S rRNA sequence. Once it binds with mRNA, then the IF2 initiation factor, that is the GTP bound factor, facilitates the association of first charged tRNA at the P site of smaller subunit. In case of prokaryotes, the charged tRNA used is F-met tRNA. Now this complex of IF1, IF2 IF3 along with F-met tRNA is together called as 30S initiation complex. Now this complex formation will lead to certain conformational changes which helps in releasing the IF3 initiation factor. This will in turn cause the assembly of larger subunit and smaller subunit along with the mRNA. This also causes the GTP hydrolysis that will release both the other factors, that is IF2 and IF1. Now this complex of F-met tRNA, mRNA, 30S and 50S subunit together will be called as 70S initiation complex, which is the actual complex that helps in carrying out the translation process. After the formation of this complex, the ribosome will start moving onto the mRNA strand in search of start codon, that is AUG. Once it finds the start codon, this will get paired up to the anticodon of the F-met tRNA with the base pairs from the mRNA. Now, the F-met tRNA will occupy the P site of the ribosome and A site will be empty, which can receive the next amino acid charged tRNA. This is the diagram that represents the initiation step, in which firstly these two factors IF3 and IF1 will bind. Then it will bind with mRNA, forming the 30S initiation complex. Then the first F-met tRNA occupying the P site, and then the association of the larger subunit with the entire other complex, forming 70S complex. Second step is elongation. Elongation in turn consists of 3 steps, out of which the first step is binding of an incoming amino acid tRNA. Now, after the F-met tRNA is bound at the P site, the new tRNA will enter along with the elongation factor, that is EFPU, that is combined with GTP, forming tRNA EFPU GTP complex. This will come and get attached onto the A site of the ribosome. Once this happens, the GTP will get hydrolyzed and the GDP EFPU complex is released from the ribosome molecule. Now, the new tRNA that is carrying the amino acid will come and get attached onto the A site as shown in the diagram. Now, the second step of elongation is peptide bond formation. Once the new tRNA comes and attaches into the A site, the peptide bond is formed between these two tRNAs that are present in A site and P site respectively. The amino acid that is present in the A site will act as a nucleophile, and this will help in displacing the amino acid of the tRNA in the P site to form a peptide bond. The peptide bond. Thus, enzymatic activity is catalyzed by an enzyme called as peptide transferase. Now, the last step is translocation. This is the final step of elongation cycle. As the ribosome keeps on moving on to the mRNA strand, it will move one codon towards the 3' end of the mRNA each time. This will lead to the formation of E site and a dipeptidyl tRNA to shift to the P site, and this will lead to the generation of new empty site, that is the A site. The translocation usually requires a factor which is called as EFG, that is internal GTP bound factor that provides energy on hydrolysis and releasing GDP EFG complex. Now the new A site is situated over a next codon and it will be ready to receive appropriate charged tRNA along with the EFPU GTP complex. This is the first F-met tRNA which gets entered into the E site and this is released out. This is the tRNA that was actually present in the A site which has moved to the P site due to the translocation, carrying 2 amino acids. And this is the incoming another new tRNA carrying amino acid that will bind to the newly generated A site. The last step in translation is termination. As the elongation continues, the ribosome can reach to one of the stop codons that are present on the mRNA. The stop codons are UAA, UAG, and UGA. For these stop codons, there are no tRNA that can carry an amino acid along with the anticodon. Hence, when the terminated codon enters into the A site or when the ribosome comes in contact with the anticodon, there will be no tRNA that can carry an amino acid, and thus the termination step starts. The termination will require release factors that are of two types, in which the RF1 is able to recognize the stop codon UAG and RF2 is able to recognize UGA stop codon. The release factors help in hydrolyzing the bond between the amino acid chain and the amino acid chain and the tRNA present in the P site. This will help in releasing the polypeptide chain alone, and also the other ribosomal units are dissociated from the mRNA, leaving the mRNA strand, and thus the termination takes place. Once the translation is done, let us see what is regulation of translation. Usually the regulation of these gene expressions are controlled at a primary level in transcription, but the translation of mRNA is also regulated in prokaryotic cells. One mechanism of translation regulation is the binding of repressor proteins which can block the translation to specific mRNA sequences. Let us understand with an example of ferritin synthesis. Ferritin is a protein that helps in storing the iron within the prokaryotic cells. This is the diagram. In this, if there is adequate iron in the prokaryotic cell, the repressor protein will not be able to bind at the IRE elements, and thus the translation can happen normally. But in case of iron scarcity or iron absence, the repressor protein will be binding to the IRE element on the mRNA, and thus this blocks the translation process and thus regulates the translation. What is translational proofreading? Usually, the two elongation factors enter and leave the ribosome during each cycle of translation. These hydrolyze GTP to GDP to induce certain conformational changes during translation, and ribosomes will still be able to conduct translation, but it will be very slow and it will be inaccurate. Hence, a proofreading mechanism has to be carried out by the cell to carry out a more accurate translation. Now let us see what are the mechanisms of translational proofreading. There are total 4 mechanisms. The first one is correct codon-anticodon pairs that are favored due to higher bond stability. The initial codon-anticodon interaction in the A site of the ribosome depends on the EFPU factor, which is GTP bound, and the amino acid tRNA. If there is a proper pairing between codon and anticodon, it will cause a normal pairing and it will have a higher bond stability. Whereas if there is unusual pairing, it will cause wobble pairing, and hence there will be no higher bond stability. Due to the stability of correct base pair formation, correct codon-anticodon are favored and more tightly at the A site binding happens. What is second mechanism? 16S rRNA binding to correct codon-anticodon pairs triggers a conformational change in the ribosome. So what happens is, in order to access the accuracy, the 16S rRNA in the smaller subunit of the ribosome will usually fold around the codon and anticodon. Once this happens, and if it is a correct match, the rRNA will bind tightly, which will lead to the conformational change, and this will trigger the GTP hydrolysis. Once the GTP gets hydrolyzed, the EF2 will loosen its binding with the amino acid tRNA, and this will help in proper protein synthesis. But if there is an incorrect pair matching, it will not trigger any conformational change, which will not cause any hydrolysis, and this will block the protein synthesis as the ribosome just falls off the mRNA strand. The third mechanism is correct codon-anticodon pairs will move into position on the ribosome sooner than the incorrect pairs due to stronger interactions of the correct pairs. Before 16S rRNA acts, the amino acid anticodon will interact with the codon of the mRNA, but rest of the amino acid tRNAs are slightly tilted. That means the correct codon which is interacting is present in a correct position, whereas the other codons are always present in the tilted manner. Once after the interaction, the EFPU hydrolyzes the GTP to GDP that dissociates amino acid tRNA pair, and due to the stronger interaction, it will be able to move to its correct position from its tilted orientation. Once it is moved into its correct position, the proper protein synthesis can take place. Whereas in case of incorrect codon-anticodon pairs, there will be weaker interactions, and this will cause more rapid dissociation from the ribosome after the release from EFP. Mechanism 4. The release factors conduct the premature termination of the flawed proteins. Sometimes what happens is an incorrect amino acid may get incorporated into the growing polypeptide chain, which may lead the misreading of the polypeptide chain. As a result, there will be incorrect interaction once the amino acid tRNA carrying the correct amino acid moves into the T site. And this will lead to the premature termination of polypeptide chain. Whatever flawed proteins are terminated are released for degradation and for further conserving resources. Thank you.